At the time of Hershey and Chase's experiment, it was widely accepted that DNA was the substance that contained the genetic material in bacteria. Their hypothesis was that viruses store their genetic material in DNA as well. In their experiment, Alfred Hershey and Martha Chase used T2 bacteriophage and E. coli bacteria to prove that it was DNA. A bacteriophage is a virus that only infects bacterial cells and is made up of viral DNA enclosed inside of a protein coat. During the infection process, the phage will attach to the cell wall of E. coli cells and inject its DNA into the cytoplasm. The cell then replicates the viral DNA and uses it to make new phages that eventually destroy the cell and then leave to infect other bacterial cells. At the time, Hershey and Chase had hypothesized that it was DNA that was entering the cell and causing new phages to be produced. In order to identify the substance that the phages transmit to the cell, Hershey and Chase prepared radioactively labeled phages with a radioactive isotope of phosphorus in the DNA or a radioactive isotope of sulfur in the protein coats. The radioactivity would allow them to trace the DNA and the proteins separately throughout the experiment by measuring the radioactivity. In two separate containers, they infected bacterial cells. One container had only phosphorus-labeled viral DNA, and the other container had only sulfur-labeled viral protein coats. After infection had begun, the cells were placed in a blender in order to separate the bacterial cells from the external protein coats. These drawings so far have not been exactly to scale. In reality, the difference in size between the phages and E. coli would be more like this. This is why, in the centrifuge, the cells settled towards the bottom and the protein coats remained floating in the liquid. After centrifuging, they found that the radioactive sulfur mostly remained in the liquid. This indicates that the viral protein coats remained outside the bacterial cells. They also found that the radioactive phosphorus remained in the bacterial cells at the bottom of the centrifuge tube. This indicates that the DNA entered the bacterial cells. This means that the mystery substance that gets transmitted into the E. coli and results in new bacteriophage production must be DNA. This was their conclusion. This discovery reinforced the prior work of Griffith that DNA is the genetic material.